Thank you for joining us today, everyone. It's my pleasure to introduce Scott George, our presenter for today on the topic of the soils of the MSD pipeline path pre and post the construction disturbance. And so with that, I'm going to have Scott come up here and join us, but I'm going to first switch to sharing the screen. Yourself an introduction.
Hello, everybody. My name's Scott George. I'm a geologist really by trade, but I switched over and done a lot of ecology work in my days. Still not sure what I'm going to do when I grow up. So I've been playing around with that and did a lot of volunteer work here at one time. Haven't been coming around as much, but got involved in quite a bit of soils research here at different projects, which I'm going to touch on several of these. I've also got a lot of slides that I'm going to run through pretty quick. Some of them that are just base maps in case somebody wants to look at this on their own and identify where these samples were collected and things like that. This lists all the different studies we've done out here, even starting back in 2015. We did the baseline in 2019. We did a Lyme impact study. Some of these results were compared, some other studies that were done here were by Bailey Sudemeyer. She did something looking at your prairies and other prairies. Then I did the bottomland forest microbial study, which really is a herbicide study. Um, and then the last one is phase 1 and 2 comparison. This just shows you the location of where the samples were collected. It's about 5 acres of totally disturbed ground, maybe a little more. But we just went along in sequence and tried to sample the different communities that are present. There's new 3D maps to be replaced that has the same points on it. You can really zoom in here and see actually old furrow rows from the crops. This is just your different community types and where we collected our samples from. And we tried to get representative samples from everything along that corridor. And we can't do a soils report without at least one soils map. Everything down this is in the bottomlands, the areas we've been studying. It's all the Wilbur, silt, loam, and all these other soils around here. The Menfro and Winfield, they're all windblown loess soils. The loess in this valley's been washed in from the surrounding hills. So that's where everything starts with the soils here. It was St. Louis and the surrounding area is covered with a thick layer of loess. It's windblown silt from the Ice Age. We get up to 100 feet near the rivers of this windblown loess if it's not eroded. And then underlying all that is your St. Louis limestone. Then I think some of this is going to be insulting to some of you because you've studied this before. But there's a typical horizons in the soil profile, A, B, C, and D, and R down there. We really went down. We classified them as A, B, and C, though there is some variations on that, which I'll get into later. And soil's not supposed to be dead. It's supposed to be alive with all kinds of critters in it. And that's just an idea that there's should be something living. And then there's a whole host of parameters you can do to do soil health. If you go online, there's everything from chemical health, cation exchange capacity, soil structure, grain size, water retention, and biological diversity. So there's a whole lot of parameters you can test for and a lot of different methodologies. Being in Missouri, we went with MU Soil Health Lab and ran these parameters. Um, these are run a little bit differently in the Soil Health Assessment Center compared to their normal soil testing lab. Um, the soil health of the testing lab uses more of a water soluble nutrients and things so you can get a better idea of what's actually available to the plants and the microbes out there instead of something done with an extraction. So there's some variations there. So as you can imagine, we have a lot of data available. And this is a typical profile, actually collected over in the East Woodland, showing a darkish gray, grayish brown silt loam soil. That in this case, we said about 8 inches for the A horizon, and then down into we had a B and C horizon that we tested. So that's pretty typical of what you see around here. So the color will vary quite a bit with moisture content. Some of them do vary a little bit. This is in the North Prairie, where it's a lot lighter gray soil. The North Prairie is distinct from some of the other communities due to pH and some other factors. But this gives you kind of an idea of how much they'll vary. One of the things we found out in the North Prairie was there was a lot of redox modeling down in the B and C horizons, which indicate it's borderline wetland there. But that's not another common feature you'll see in the deeper part of the soils. Then also soils have structure, and this will get into our water stable aggregates and things, but there's pore holes from roots and worms and burrows. Here on the lower right, can you see that? Nope. Anyway, or on the lower right there are some of the burrows that were highlighted by having a nice lime mud infiltrate into them. You see that? Yeah. Well, there's some of the burrows and things that are in there. In this case, they're uh, covered with lime mud with some good sulfate levels in it. And the other plot up there in the upper left, the dark colored one, that's from the right outside the cabins here from the herbicide study. Some good rich soils. So most of us would look at that and say, at least the one on the left looks like pretty good soil. There's also a few areas in here that you actually have redox modeling. You have in your forested wetland over here, the iron goes in solution and precipitates back out, and you get this redox modeling. So this is all in the Wilbur silt loam series. And there's a standard reference protocol up in here from some farm field in Indiana or something. But 0 to 7 inches is their AP for plowed. And then they have a weekly developed B horizon, 1 and 2, and then a CG horizon. This is from Indiana. Our descriptions varied a little bit from that. Down in here, we tended to go, this is North Prairie, we done A1 and A2, and then a B, and then a C horizon. Our NRCS soil scientist that did the monoliths here, he used a different system. So depending on how you're listening to, you'll occasionally get some different classifications on these. One of the first things we did is looked at grain size we collected a lot of data in 2019, then we went out in 2022. 2019 was a much easier 2022, we had a lot of rock and rubble. But you can see the comparison here just on, I took the A horizons from phase 1 beforehand, and then the A horizons from after. And the only really big change was an increase in the sand content. That's up over here on the right. You went from about 8% to 20%. Clay content stayed about the same, but then the silt content lowered some. So there was some actual change in the grain size, but not enough to really affect plant growth, I think, in most cases. But if you plot those, the green arrow is where this plots on these soil diagrams based on the percent silt, clay, and sand. So number 1 is phase 1 before, and then number 2 is afterwards, and they're still both classified as a silt loam, just more sand in the second phase. Then we all know that soil is supposed to consist of, mineral particles, air, water, also organic matter and roots and other things like that. And then how those different parts are available in the soil is the bulk density, and that makes a big difference on plant growth. So this is how we collected bulk density from before, when we have quite a bit of data in actual soil. Then when we went out there in phase 2, we ran into lots and lots of rock. We actually destroyed this bulk density tube, and we had to buy them a new one. But there's small rocks in here. There's medium-sized rocks here, and then there's really big rocks we had to move out of the way just to get the holes done. We even tried renting a power auger and things, but it didn't go too well. And a lot of the people in the ag field, a lot of what they go on is bulk density of your soil. And there's quite a few programs in NRCS where you're required to collect this data, and it'll give you some idea on the health of your soil. And there is some criteria here we have. This over here in the far left is the bulk density of the A horizons in the mean value from phase one. And I seem to be losing my cursor here. The green line is anything below that is ideal for plant growth. So your phase one soils were great. Up here is where it's supposed to restrict growth. If you look at the bulk density of just the soil fraction in phase two, it's higher, but not all that high. But again, we just were testing the soil part of that. We weren't looking at the big rocks. So if we assume 30% rock in a typical profile and a bulk density of 2.7, then you get up here past the restricted growth. And then on the far right here is if you look at just the rocks themselves, they obviously have a very high bulk density and very low porosity and very low water content. So if half your profile is rock, you're looking at quite some density and very hard to get anything growing in it. Then also your porosity decreased quite a bit, but this is also excluding the big rocks. Then your water content over here on the phase one, it's gravimetric and volumetric are just the two differences here, but you can tell in your phase two soils, it just dropped. It was astronomical. They didn't hold any water at all, hardly. So that's looking pretty poor. And then the water stable aggregates is how much those clumps form with roots, and the fungi is really important in that. And that'll tell you how well that soil will be crumbly or friable and breaks up into chunks that allows water and nutrients to pass through. And so you're definitely a significant drop here. So much more soils. These are the physical parameters. Um, and then real briefly, I just wanted to touch on this. We have a lot of information, again, from the different sections. Your bulk density increases with depth. As you go from the top soil, you can see over here, we've got A, B, and C. As you drop down, the bulk density increases just almost uniformly in every profile. And then your water content also decreases, even though the soil will look moister. The ability to hold water decreases with depth. And then also the porosity decreases with depth. So that was very nice, repeatable information and very consistent across the site. The same thing goes along with the profiles from nutrients and the nutrients in carbon and other things that are available. The A horizon has the highest numbers. If you look at it, then you go down to the B horizon, it's lower, and then the C is the lowest. And the same with the active carbon, the phosphorus, your nitrogen, and then the plant available nitrogen. So it's all pretty consistent story. And we saw none of these trends in the phase two data because all the soil had been taken and mixed up together and was the same all the way through. Before I leave phase one in the soils, I had mentioned that the North Prairie was a little bit different, I think because of the ponding of rainwater out there. But if you take of all the phase one North Prairie pHs along with our lime study samples, your pH is in the blue over in here, below 6.5. And then if you look at all the other phase ones without North Prairie, they're much higher. So there is quite a bit of difference in that section. That was confirmed by other studies. And then I threw some of these in here in case anybody asked me serious questions about some of these parameters. The cation exchange capacity is the sum of the negative charges on there. And that determines how much the nutrients can attach to those negative charges and then be released for plant growth or microbes. So it's a very fundamental property of the soils. So it's the sum of the base cations and some of the acid cations, which they estimate off of the pH. And then looking at comparing this is the base saturation. This is the sum of our base. And I've changed the axis on here a couple of times. So these are the phase 1 soils in blue, phase 2 soils in orange across here. The units vary so much between these parameters, I had to go with some individual graphs. But as you can tell, and there's a huge jump in calcium, as you might expect when you crush up a whole lot of limestone and mix it in with your soil. Not much change in magnesium, not much change in potassium. Our soils around here really don't have any sodium to speak of to leached out. So the sum of your base cations is skyrocketed also in this sample. And then that, because you have so many cations in there, so much calcium, it lowered your cation exchange capacity. So you have less sites available for nutrients to be attached to. There was also quite a jump in the pH. It's measured 2 ways, with salt and with just water. And both of them, the phase 2 is much higher. And that's to a large part due to the calcium in there. But if you look at an overall pH increase of about 0.5 to 0.6 units, that's quite a bit different than what you guys had beforehand. And can make a difference to some species of plants. Again, your percent base saturation skyrocketed also. You were near 100% base saturation here before. Now you're at over 300% base saturation. So you're super saturated with bases. So I'm thinking limestone glade. But also, yeah, no. If I was just looking just at the soils data, I would say try to plant a limestone glade or limestone woodland, something like that. But you're in a floodplain, so it's going to be hard to keep a glade out there. Your total nitrogen dropped astronomically. The mineralizable or available nitrogen dropped dramatically. And the same with some of these other parameters. Total organic carbon presented, and then the active carbon, maybe not quite as big of a decrease in there, but much lower levels. Again, these are the A horizon mean values from the different units. And then plant available phosphorus also dropped quite a bit. There's another way you can look at this statistically, in which we were starting to do for the report. But I'll just go through this one briefly and not bore you too much with them. But these are the whisker plots, which show you the range of all your values, the maximum value and the minimum. You've got the mean value here in the center. The upper line here, top of the blue box, is the mean of the values above the mean, total average or mean, and this down here is 25%. So this is about 25% of your data. This is about 50%, and that's about 75. So you can get an idea of where all your values are falling in. And if you look at the calcium in phase 2, all the values, even, you've got one outlier here, but basically all your calcium values were way above the phase 1 values. And you can do that for every one of them to give you an idea of how the statistics fall out, but that would be another long, boring conversation. So what does all this mean? It means you have nutrient poor soil. Yeah, I thought maybe that would sneak in. And there are some actual guidance you can get on this, on what is a good number and what's a bad number. In some cases, it's not straightforward. But this case, in saying phosphorus, you've got to convert the units to pounds per acre instead of parts per million. So you multiply it by 2. But your values come out into being very low range. When you look at nitrogen, they don't do an actual most of the soil tests don't do total nitrogen. They base it on the organic content and your cation exchange capacity, assuming if you've got a lot of organic content, some of that's going to be available nitrogen. But you're pretty low on the scale here too. You had an organic matter content in phase 2 about 0.7. And then this gives you an idea how much fertilizer you'd have to add to bring that up to par. So there are some you can actually go online and enter this information. Remember, it's a phase 2 A horizon means. I put in a wildlife food plot with a yield of 0, and then they'll print out a recommendation of pounds per acre of fertilizer. And so just based on this, they would want you to add a lot of nitrogen and phosphate and potassium to the soil. This part might be a little more interesting and a little less cut and dry. But we all know a healthy soil is full of all kinds of bugs and critters, fungus, bacteria, nematodes, and all those things are very important to plant growth and in fact, the mycorrhizal fungi, that's really a hot area of research, both for agricultural fields and for prairies and restoration. So there's a lot of biology going along in a healthy soil. And what we did is we used the phospholipid fatty acid tests from the MU Soil Health Lab. And they were really some of the first ones in Columbia, University of Missouri, Columbia. And they're pretty early on in their research on this and making this testing widely available. They're kind of a leader in that. And in essence, it's part of the cell wall, and there's different structures to these different filaments and things, different saturation, functional groups, and they all break down right away after they die. So when you're testing the soil, you're getting an idea of what's there, living there now. And we do the arbuscular mycorrhizal fungi, AMF, the gram-positive, gram-negative fungi, the acetinobacteria, and then other fungi and eukaryotic cells, which are the bigger bugs and bunnies and nematodes and things like that. So those are the results we got out of that. And then on the quick memorize all this. But this was the more fun part of it. Do some research. There's a lot of research out there available on it. And this fungi, the AMF, actually penetrates the roots. So part of the fungi is in the roots and part of it's in the soil. And it's a symbiotic relationship. The AMF the plants provide the AMF, the fungi, with carbon and hydrogen for energy. And then the AMF provides the plants with nutrients and increased stress tolerance, drought and disease. It also has a big factor in forming this water-soluble aggregate, soil structure. So they're very important in that aspect too. Then gram negative, gram positive. I'm not going to go into the details of the structure, but there are some differences. Gram negative bacteria is more directly involved in plant growth. And that's one of the ones that use the legumes that'll increase the nitrogen in your soil. So a lot of people really, if you're interested in just agricultural crops, people want high gram negative concentrations. Gram positives are closely tied to the rooting zone and dominate. They're widely dispersed, tend to decompose organic matter. So they're more decomposers. And then just the regular fungi, there's a lot out there besides AMF. And they're mostly there for breaking down organisms and decaying things that are like that. Acetotin bacteria, there are a variety of gram negative that are used in decomposition, but they also form the antibiotics and the antifungal compounds that help the plant survive stress and disease. So that's another important parameter. And as you may have guessed from looking at this soil, that there was quite a bit of difference in the biological life between in the soils between um, our control group over here on the left, and then over here on the right is the disturbed corridor when we were doing the sampling. One of the things you have to do this sampling, you can't do this biological sampling once and then try to do a different segment or a different test cell later because it depends quite a bit on soil temperature and moisture content and other factors, time of the year. So you have to collect them all at once. So we couldn't collect beforehand. We could just collect some in the corridor that was disturbed, and then we went outside to look at those natural communities, um, and then compare the two. And if you can tell from this, um, the blue dots are the impacted zone. This is the total PLFA in per grams of soil. The blue is the impacted corridor. The orange is are the controls. So you can see there's quite a difference. And actually doing detailed comparisons across these is kind of pointless because you can tell that there is a huge difference in the total biomass of the soils, as you might anticipate. But one of the things I did find out, I was looking at the control here on the left. This is in the prairie. The impacted area is on the right here. And if you break it out as a percentage of that biomass that's there, they actually came out pretty similar. I think over here, the gram-negative bacteria was the biggest one. It was 49 in the impacted area and 47 in the control. And so most of these look overall, so you have a lower biomass there, but it's about the same composition other than your eukaryotic cells, the big guys, the bugs that crawl around in there. There's zero in the impacted zone. So that was a big difference between his total mass. Uh, yeah, there wasn't much living out there, given the organic content and all. On these, and most people collect them 0 to 3 inches or so when we were doing the soil sampling, we used a hand auger. But when we were collecting the biological samples, we used these trowels that all had to be decontaminated and things between, because you're collecting biological samples and they also have to be refrigerated right away and shipped overnight. So again, there was an obvious difference between the impacted soils and not, but we did collect quite a few different communities in the control. Some of it was prairie, some of it was woodland. Number 3 here was underneath a rotten log. And as you'd expected, that came out to be real high in total aggregates. And then over by the boring 8 near the Litzsinger Road, we collected our control in basically a lawn area with sedges. So there was some variation in the communities we tested. So if I plot that, if you take out the main groups here, the fungi, the eukaryotes, the AMF, the gram-negative, pseudomyines, gram-positive. So if you look at the composition of these things from the different control groups, there is some variation that you can attribute in some cases maybe to the natural community. The one here at the very center here was the one collected underneath the log. And we knew that was biased, and that obviously has a lot of almost every one of the parameters is high. But then it dropped off when we get down here. We called it the wetland swale. There wasn't any wet. Number 8, that was the area with the sedges and the grasses, and that's pretty low. And one other thing to note in here were the North Prairie. We collected a duplicate that was about 4 feet apart. I wanted to get some idea of the variation in the communities in the North Prairie. We did this with the nutrients also, 3 or 4 feet apart. As you can tell, there's a big difference between my duplicates. And I think one of them was, I'm not going to go, maybe I can go back here real quickly and show you. Uh, This sample was collected, the high number on the North Prairie was collected underneath a lot of shaded forbs where the soil was sort of moist and a lot of diversity here, where the duplicate was collected over here somewhere in a sunny, dry soil with just sedges. So even within a small area, depending on your moisture content in the vegetation, you can get some variations. Um, and I'm not sure what else we can say here. A lot of the same groups were there, but just at varying concentrations across the site, which I think ties into vegetation and moisture. Another thing they're really big on this is looking at ratios between the fungi and the bacteria, the predator prey, the gram negative to gram positive, saturated and unsaturated chains, gram-negative stress, and the mono and poly. One of the problems you run into with just the totals on looking at the different communities and looking at these ratios is they vary widely. If you start looking at the criteria set up for a cornfield, you guys are flying, at least you're every natural community we sampled out here was way above anything in a cornfield, obviously. I think even some of your impacted areas came out above a cornfield because you're not applying herbicides and things. Um, One of the interesting things, our highest concentration sample seemed to have a lower ratio of at least of these mono and poly groups, which probably means it's more balanced. So overall, the lower the ratios, the more balanced your communities are. This gram negative stress is the gram negative over the gram positive. Um, and I've heard both things. If you have high gram negative stress, it's a bad sign. But MU says, no, that's a good sign because a higher gram negative stress means you have more of the gram-negative bacteria, which are helpful to plant growth. So a little that can vary. And then here's just some things I dug up on the web. Again, most of these people are looking at agricultural fields and things. Uh, but certain ratios were listed as good or well balanced or something like that. Um, but the MU Health Lab were saying that higher fungal bacteria, predator prey, saturated, unsaturated ratios, and gram-negative stress are better. And lower the gram-negative ratio, the better. So I can give you some idea of where some of these come out in absolute values. Um, but again, as long as your comparison is a cornfield, it's a little difficult. How are we doing in time? So I put most people asleep.
You're doing right. We got 40 minutes in.
Okay, well, probably we'll have some time for questions, I think, then. Um, One of the other things we did here was a herbicide study, which was right out from the cabin here, where it was covered with it was a monoculture of evergreen, at least on the ground cover, and then you guys treated it with some herbicides, uh, and then planted some natives there. So in that study, we looked at, these are our three impacted samples here. Evergreen was the ground cover. And then we did two controls, one of them with mostly herbaceous growth and one of them mostly with trees and things. This evergreen, this plot 3 here in the middle, I labeled that swale because it was in a low drainage swale area and we hit a pocket of rich soil. Um, so if you wanted to um, knock this one out of there, just forget the swale for a minute. Your evergreen ones generally had lower biomass and lower group values for these different groups than your controls did. So one just looking at this data, one could argue that the native plants are increasing your microbial communities.
Over the invasive winter creeper, correct?
Yeah, oh yeah, compared to the winter
It's a
creeper.
very shallow rooted plant.
Yeah. right. And but also there are some differences in the landform a little bit. But overall, I would say that that's an indicator of the of lower biological community diversity in native plants. I did sort of the same thing here, trying to look at did the different groups vary, looking beforehand, phase one. Again, this is the herbicide study in phase two, but overall they came out pretty close to the same. Though there were some differences here, like with the gram positive and the green very, oh, wait a minute. That's, anybody gone colorblind? Oh, okay. Anyway, there were some slight variations in differences in between the control and the post sample. And then I'll look also looking at the different plots, just trying to compare these values a little bit better between, oh, I've changed the axis on you. This is phase one, phase two. When you look at the evergreen ones, ignore the swale for a minute. In the evergreen area, after you killed it, and so we waited a full year for some regrowth in the natives. Actually, your total biomass went up in those after treatment. So that's a really good sign. And in our controls, it went down. So that would tend to make me think that maybe there is something here with killing off the winter creeper and letting the natives come in. Maybe there is some increased biological activity, especially given with the controls. Again, just a comparison of the main groups. And some of these, this is just plot 1A. Yeah, again, looking at before and after phase one and phase two, there is um, there is an increase in these different groups after you guys killed the evergreen and planted the natives. Over here, your eukaryotes, the bugs went down, your total fungi went up. So you may have killed off a few of the bugs. But overall, it looks to be an improvement. It certainly hasn't hurt anything. Uh, One of the questions I had when I was working on this was, what does this compare to other prairies around the area, and that's what Bailey did. And she got some information from Bluebird Meadow, which was a young prairie just under starting restoration, north, south, and pasture prairie, and then the Calvary Cemetery, which is considered a remnant. I was up there for some collecting those samples, and it was pretty good looking soil. But in general, your comparison, if you look at calcium levels, you're pretty well similar. Just on the organic matter, you're pretty similar, other than Calvary, that remnant prairie. Same with potassium, fairly similar. But again, Calvary kind of stands out. So I can kind of give you an idea on how it compares to the rest of the world. And I wasn't sure if we'd have time, but I'll go into it anyway. We did the lime impact study where they were doing tunneling and they ground up a lot of limestone and then sent it over your way into the North Prairie. So we had a layer of lime mud covering some places, and we went in and tested that. And I haven't done, I don't have a real good comparison of the microbial populations here, but you can tell one of the things that happened is the lime mud shut off all oxygen. That's why you had a lot of wetland plants out there, at least initially. It also then caused, because there's no oxygen, it causes reducing conditions to create these iron deposits. So it was very distinct. None of this was there before. Um, and I guess you guys will keep growing in that stuff. I mean, what you're going to get to grow.
Well, a lot of that was removed in like over the next couple of years of them working.
Plowing through.
Yeah, so some of that was remediated over the length of the project. but we wanted to make sure that we had the impacts just in case.
That's all that stuff they dumped.
Yeah, that was all the mining slurry that was dumped into the North Prairie.
I don't think it was all intentional.
It was carelessness.
Yeah, it was pretty bad. We got some interesting results on that. One of the things we were worried about was sulfate. So I broke this out. The lime mud had lots of sulfate in it. The soil below the lime mud had fair sulfates, detectable levels. Kind of ignore this 1/4 sediment that was from flood water, and it's kind of an outlier. I'd have to talk around. And then compare that to your background sulfate levels in the North Prairie, which are almost non-existent. So you're really comparing this background level over here to the lime mud over here. And it's pretty distinct. You can make it anyway, just pick your units right and you can make anything look distinct. But no, this case, it's amazing how much of that happens. So we plotted it on a map just to kind of get an idea of the area of impact. Again, the sulfate levels over here, 3, 5, over here, 2,300, things like that. So this is, if you're looking at a contaminant plume, this is kind of the standard stuff to draw out with. And then obviously we were worried about calcium levels. And in your background, they're down in this range. Lime mud is obviously a big problem. And also the soil underneath the mud got a fair amount of infiltration in there. And the sediment, I think, is partially due to periodic flooding. We had a flood when it was still out there and complicated matters. And we plotted calcium levels also here also. Uh-oh, we're going. And really that's all I have. If you have any questions about all that simple stuff.
I'm curious if you measured chloride applied to the soil in the
Please repeat the question for the audience too.
Well, the question was looking at chlorides from the road runoff and salt impacting the soils. And we didn't actually get a chloride number, but we got a sodium number, sodium chloride, road salt. And we only got sodium detections on the sample. It was right next to the creek in phase 1. It was the only detectable sodium. In phase 2, we got some reported detections, but they were right at the detection limit, down in the parts per billion range. And most of our soils around here are too leached out to have sodium. So it did look like the creek flooding probably did impact some soils in the surrounding area.
Yeah, my question was, are we going to have to amend the soil with more organic matter before we plant, or
that depends on what you're actually planting. I mean, most people would recommend you would add fertilizer in there, but I'm not sure that's something you want to do, given especially the compaction of the soil and how it would be hard to get it to infiltrate the soil. It'd run off.
Answer that is not to the audience.
okay. Oh,
And that's
What?
Because they're not going to be here for like going out looking at the back. So you're just going up there and repeating the question. And
okay.
So you have to add nutrients, right?
What was the question?
You have to add nutrients to the soil.
or any kind of amending.
Okay. What do we need? We're going to be here with this profile that doesn't look.
Okay.
And
Oh, hello, all the Zoom people and people in the audience. I am Katie Sims. I'm the restoration coordinator for the Project Clear MSD path. So the phase 2 part of what Scott was talking about. And so I'm in charge of reconstructing native habitat introducing plants back into that soil. So that is a challenge. And so what I've came up with and have talked with James and just gotten feedback from others, and is that so far we have seeded onto the path native plants and early successional plants. But our strategy that we're using to get plants growing and so that they can actually grow and break up the soil, we haven't added anything to the soil, is to use plants that are disturbance tolerant, and will likely grow in that kind of soil and not so much of a pristine prairie kind of habitat. Another thing that we did before even seeding native plants is seed cover crop over it. And that was to try and add nutrients to soil, break up the soil, get an organic layer going, help with erosion, things like that. So we put a cover crop that went down in fall, winter of 2022, put a second round of cover crop on the whole path in spring 2023. For the most part, that did okay on the path. There are some parts, especially here by the driveway, where flooding is more common and frequent down here. And so that wiped away a lot of that cover crop and seed. And so it didn't do as well down here, but it did pretty good in other places. And then starting in October, we did the first native seeding, and then in December, and then a couple weeks ago, we finished that up. And so just seeding in things and seeing how they take is the strategy.
What about tree replacement?
So
Where did that start?
So we had a question about tree replacement, since a large part of the eastern section was bottomland woodland and the southern section of the pipeline path. So first, want to try getting some prairie species and early successional stuff, herbaceous plants to grow before I try and plant any trees. Except for this year, we will have a few trees put in here, and we're going to make this section by the driveway into a savanna. The other two sections, so that northern section will just be continuation of northern prairie, and then this section that is dissected by the creek on the eastern part of Deer Creek is going to be prairie as well. But this savanna section, we'll put a few trees in there this year by some contractors. But as far as any plantings on the eastern section for that woodland, I'm going to hold off until a couple years and then see how things go. Yeah. And also, so there's also a, what is that called? What am I trying to think of? The 15-foot, 14-foot, yeah, the easement. Yes. So there's a permanent easement over the pipeline. And so we can't, so MSD can get in there anytime they want, basically. Yeah. Yeah. If they need to, it's their pipe. So we can't plant trees over the pipe. And so it will never be a woodland like it was again, unfortunately. But
I know it's sad that we lost so many trees and we lost some woodland, but we've gained prairie. So that's it's a benefit.
With deep roots,
yeah.
Hopefully.
Well, and prairie is a lot less common in the St. Louis area for an area that once was the Prairie City. So
yeah.
So anyone else?
Prairie, Missouri is north or south to east.
Yeah, to the north and to the west.
Into the west.
Right, yeah. If you look at the map of the prairies of Missouri, St. Louis has got quite a few prairies mapped, but as soon as you get down towards the Merrimack River, they die out and things. So this was still a prairie area for the most part. Even though NRCS maps most of the potential natural communities here, as in like deep lush woodlands and things. But it was still have included a lot of prairie grasses. Do you have anything that actually works really good at breaking up the soil? I've heard different stories on things.
So, I mean, so I've only been here for about a year, and the only thing that I've seen grow have been the annual cover crops. And so the common wheat that they put down, the roots did not go very deep on that. And then they put down Sudan grass, and that was basically corn. Like that's just milo, sorghum. And so it has prop roots, and it also didn't go very deep. I mean, it broke it up a little, and it's gonna die and create some sort of a thatch layer or something. But Then on the southern section, we had a variety of different crops put down. So some mustards, legumes, grasses, and so sunflowers, buckwheat, to try and get things that have deeper roots and have some like a diversity of plants. And so there was some radishes and things that would have deeper tap roots. And because of the flooding, and then it was really dry, and the quality of that soil was so compacted, so nothing penetrated deeply into that soil.
The couple radishes that were even there were growing on like the surface.
Yeah, like you could, they were popping out. Yeah.
Yeah, that's what they couldn't get down.
Yeah, I know before I started this, I didn't realize how important that soil bulk density was. And I mean, it's probably twice what it was before. And then the rocks, it's probably 30 times what it was.
Yeah.
So
it's tough.
So aside from the cover crops, though, the plants that were naturally growing on their own in the first year, so all these successional species that came in from floodwater, like the huge range of annuals and things, there was a lot of things that were just very depauperate, so very small, dwarfed in size. The only plant that seemed totally unfazed by the fact that the path was there was the boneset.
Yeah. So
the late boneset.
Yeah. So James commented about how the late boneset grew really tall on the path this year because we kind of let all of the early successional kind of weedy, disturbance loving plants grow on the path, kind of like a cover crop that we didn't have to pay for or seed. And so we just let it grow. I mean, we would remove anything that was too invasive. But yeah, the boneset, that was fine. It was like a sea of boneset on the east side, and it was really pretty, and yeah, it got tall.
Better than nothing.
And yeah, right. It's native. The butterflies were all over it, and yeah, so
almost every other species though that was encountered was dwarfed in some way. Even all the exotics that were popping up.
Yeah.
Well, I know the calcium levels are so high. I've heard of calcium toxicity in some plants and species, causing them to be dwarfed in size. So you may be running, you're super saturated with calcium, so you may be running into some problems there. But it's impossible really to predict for each species. Nobody's studied every plant's tolerance to calcium, the plants.
So I guess I would go to Katie instead of Scott. So if you want to answer that, I'm hearing this report, or Scott, if you want to pop a prime in there too.
Yeah. So Tom said, how does this soil report inform your choice of plants?
Weedy succession.
Yeah.
It will grow.
Yeah.
I mean, if we were not in a bottomland area, given the soil compaction and calcium levels, I'd say a limestone glade, little blue stem, size oaks, grama, and some other things that are common in a glade-like community, but you're down in a floodplain. And so it'd be really hard to keep those going forever. It might be interesting to see if a little blue stem would grow there, but
we have planted little blue stem in some of the drier areas that are outside the floodplain.
Oh, okay, on the slopes and things.
That are still disturbed by MSD, but not going
to get the Indian grass, Sargassum nutans, that'll grow in almost anywhere.
Yeah.
But then it tends to be real aggressive and dominant.
Could be better than nothing.
Yeah. I'm amazed you got that much boneset growing in there.
Yeah.
Getting anything to grow there is great.
And then there's some questions, I guess, about
I'm sorry, I can't see.
about some herbicides.
Oh, we'll switch again.
Here, you want to come up and sit down? You can probably read them right here. All right.
The question was the glyphosate? I'm not sure that's what we used on the evergreen. Adam?
It was tractor.
Right. Oh.
It was tractor.
Triclofair?
Tractor here. It's the one that she mentions first.
Oh, okay.
So yes.
Yeah, that is the one we used, and to kill off the evergreen, and that may have impacted things in the short term. We came back after one growing season and actually had higher levels in the of microorganisms in the impacted or treated area after the treatment as before. So we didn't notice any negative impacts from the herbicide use, but we did wait a year.
That's the last questions for online. Any more questions for your person?
James, I'm a big fan of old Mississippi, lower Mississippi River water maps that were made by Harold Fisk back in 1944.
Yes.
Soil studies of all the states along the Mississippi. And he was able to plot the ancient courses of the river and back and forth in multicolored soil. Is there any chance that your data have enough resolution to be able to show some courses of Deer Creek along the prairie?
No, not really. We can actually we can see with the I guess we can't bring those slides back up. But in one of the early slides, I used the 1 meter resolution 3D maps from USGS, and I can leave a copy of these slides here. But it does actually show some of the old side channels and things that we that were present. And you can, but nothing like what Frisk did on the Mississippi River floodplain. I was trying to do that for out at the Creve Lake area because I have some old historical maps and try to map the old channels, but it gets a little iffy. And I don't know if you almost have to zoom in on this to see this.
You can see it back here, Scott.
Oh, okay, maybe we can't. Well, obviously you have an old oxbow right in here that still gets flooding periodically. And then you have some old oxbows or some old channel stuff in here. So yeah, you can kind of see, and then there's cut to the drainage. This pasture berry, the berms were right in here. But this is your forested wetland over in here that might have been part of an old channel. But yeah, you can get some idea of where the old channels were here, but not real long ago. You can actually see some of the old furrows from the farming out there, and then the trails show up.
So what technology did you use to get those images?
This is it's available from the National Map Database. I do some GIS work, and now for the St. Louis area, you can directly download these 1 meter 3D maps for most of the St. Louis areas. And so they're really fun to play around. And if you play with them right, you can zoom in even further and show, we could highlight just this area in here. Now this map, I didn't put a date on it, was before construction.
So that's the topographic.
Yeah. It's showing land elevation.
And the other thing to consider too is like really just how far back can that show, because don't forget too that the railroad company back at one point in time came through and really did a big number on what happened. And at some point in time, I think it was this MSD pipe that they removed was about 100 years ago. So don't forget that MSD had already been through this area too. So Deer Creek has probably been moved around a bit.
Right. Yeah, we could go back and look at the real ancient topographic maps. That might be something. Yeah, I love this.
I'll show that to the students.
Any other questions? I think we should definitely talk about mastodon poop. No, it's part of your soil. So, I mean, anyway, no, I didn't have any questions. next month.
Yeah, a question was asked, when's the next soil test? So with the restoration happening, it might be good to be looking at the soil at like in increments of like every 5 years or so after the vegetation's had time to start adding that organic material back into the soil and maybe see some considerable change. You know, if we don't see a big considerable change in 5 years, maybe we wait every 10 years or something for sampling. But
seeds recently?
we'll be doing that when we go out to look at the path. All right, so thanks all, and lots of thank yous from the audience, Scott. All right. It was recorded, and so we will share this out. So thanks all.